Okay, I think that is everyone for now. So again, welcome everyone by today's seminar of paleoparks. Our speaker of today is Elizabeth Sibert of the Woods Hole Oceanographic Institute in the USA. And her talk is going to be about something fishy this way comes, investigating shark and fish evolution using microfossils in deep sea sediments. So first, a bit of housekeeping, the format of today's seminar, especially if you're new here. We first do the short welcome and announcements, and we're doing that right now. Then we have Elizabeth's talk, which will last for around 30 minutes, followed by a moderated Q&A of around 10 minutes. Elizabeth has agreed to stay for tea time, so if you want to have an informal chat after the talk, then you're free to stay afterwards. And don't forget, if you have any questions, send them via the chat to the questions at Paleoparks host, which today is Isaiah. And a bit more housekeeping. So Paleoparks values the participation of all folks interested in the paleosciences. So please remember to abide by our code of conduct during today's seminar. If you're in the Zoom room, you should have agreed to our code of conduct, but if you haven't done that for any reason, please have a look at our website. You can find our code of conduct there. Also, out of respect for the speaker, please mute yourself for the duration of the talk. You should be muted anyway, but if you find yourself unmuted, please be respectful and mute yourself again. And as I already mentioned, you can ask questions by chatting to the questions at Paleoparks host, aka Isaiah. You can also use the raise hand function if you would rather ask the question in person. And also, if you have any technical issues, you can also ask the questions at Paleoparks host. We have also built closed captions into Zoom. They should be enabled, and you can use the CC button to either show or hide them. And then finally, we're always looking for more speakers. So if you know an outstanding early career scientist, or you are one and you have a great story to tell, then please nominate a colleague or yourself in this link. This link will also be dropped into the chat shortly. And then finally, we're also always looking for weekly feedback from you, where you are from, how you identify, just because that helps us improve what we do at Paleoparks. So if you want to fill out the weekly feedback form, anonymous, completely optional, you can also do that. Then a bit more about today's speaker, Elizabeth Sibert. Elizabeth did her bachelor's in biology in San Diego in the USA, followed by a master's and a PhD in biological oceanography at the Scripps Institute of Oceanography, also in the US. She did several postdocs, one as a junior fellow at the Harvard Society of Fellows at Harvard, and then 2 at Yale, first as a Hutchinson Fellow and followed by an associate research scientist position, also at Yale. Currently, she is an assistant scientist at the Paleofishes Lab at the Woods Hole Oceanographic Institute. Elizabeth has also been a past member of the Paleoparks Steering Committee for several years. And we have as a tradition that if you are an outgoing committee member, you also give a talk at Paleoparks. So that is this occasion of Elizabeth's talk. So yeah, without further ado, I would like to invite Elizabeth to take over the screen and share about her paleofishes.
Right. Well, thank you, Nina, for that absolutely lovely introduction. I have to say it is absolutely wild being on this side of the Paleoparks seminar. Usually I'm on the other side, and it's been a real privilege to see you all take this thing and have it grow. So today I'm going to be, as Nina said, giving this talk titled Something Fishy This Way Comes, Investigating Shark and Fish Evolution Using Microfossils in Deep Sea Sediments. And for today's talk, I'm going to just sort of give us a little broad overview of what I'm thinking about, and then share some more interesting stories that I'm thinking about more recently as we go along in research. As mentioned, I was a Paleoparks committee member, and I just want to highlight that a little bit because it was one of the most absolutely important things that I think I've ever done in science, and I really am thrilled to see it continue even as I've rotated off of the committee. And so just a wee little bit of history, if you'll indulge me, is that almost a month ago, or almost 5 years ago to the day, was when we came up with this idea for this seminar series. It started out as this little idea to address the isolation that all of us were feeling, having been suddenly sent home to work from home alone and not having any kind of departmental structure or institutional structure to connect with each other. And so I actually was chatting with some friends and we are like, hey, maybe we could do something that would bring together the early career community in paleosciences. And in early May of 2020, I sent an email to some friends being like, hey, you want to try and pull something together? We developed a diverse committee and had our first committee meeting in late May. And our first seminar was less than a month after that. And this thing has been going since then. My personal service on the Paleo Perks Committee went from the beginning, May 2020, until I rotated off in September of 2024. The jobs that I did, I was the outward-facing communications in email list manager. So for all of those years, if you got Paleo Perks seminar email announcements, that was me trying to figure out how to do that. So it was 2 to 3 announcement emails per week for about 180 seminars, and we had an email list of over 2,000 people. One thing that I learned during this time is that email lists are really quite a challenge to manage, especially when they get to thousands of people. And we ended up seeing this go through about 3 different email server migrations until we landed in our home server, where I hope that we will be able to stay. In addition to doing the outward-facing communications, I was a committee coordinator, so I did a lot of organizing what the agendas for our meetings might include and facilitating meetings, and I served as what I called the catch-all. So I kind of knew approximately what people were up to and tried to keep track of all the bigger picture things so that everyone else on the committee could focus on what they were doing as individuals. And so, like I said, I was around for quite a while, but I rotated off about 9 months ago. And it's been absolutely a privilege to see Paleo Perks continue. Almost the entire committee right now is not anyone who was part of the original committee. We have one person still left, but everybody else is new. I'm talk number 199, which is just so cool. There's over 3,000 people who have signed up to the mailing list with about 2,400 active email list members, which is, again, really quite amazing. We've had speakers and audience members from dozens of countries and held seminars and events. And there have been 14 committee members over these years. And hopefully maybe one of you will become one of the next ones. And it's been a real privilege to be able to contribute to this organization that started off as a little thing we were trying to address COVID isolation. It has really grown into a global community. And so I just wanted to thank the current committee for continuing this little legacy and thank all of you for engaging in this. It's such a privilege to have seen it start, and I hope that it continues on into the future. So thank you for letting me be a little bit sappy. Now I'll actually jump into some of the other things that we're thinking about. The other thing I want to mention just before we get going is that in addition to the work that I've done in Paleo Perks, I do a lot of work in accessibility and STEM. And as I transitioned into my faculty position, I actually had the privilege of building a lab space. And so my lab is actually the first wheelchair accessible and ADA compliant lab. ADA is the Americans with Disabilities Act. It basically is, in the US, a way to ensure that spaces are more accessible to people with disabilities. It's the first lab of that nature at my institution. And so if this is something that you are curious about, I'm super passionate about this, and I'm happy to chat with people about what kinds of things you can do to make a lab space more accessible. And this is something, if you want to stick around for tea time, I'd love to chat with people about that. All right, so enough of this. Let's get into the science. So I like to think that every scientist has a big question that's driving them. Behind all of the moments that we spend at the microscope, behind all of the little teeny tiny data points that we're collecting, behind all of the really tedious tasks that we have to do in order to get to our science, we all have these big questions. And for me, one of my big questions is, how do fish and marine ecosystems respond to global change? And I think this is a huge question. I think, like, hundreds, thousands of researchers all over the world think about questions like this, and we all come at them with slightly different perspectives. And so you saw in my bio that my background is in biology and biological oceanography, but yet here I am sitting in a paleo lab in a paleo seminar. And that's because I come at this question of this very biological question from a paleontological perspective. And so that's what we're going to do. That's what we're going to explore today. And I just want you to keep this question in mind as we go through the rest of the talk. So I think many of you in here probably already care about fish, but I often give talks to geology departments and they're like, why do I care about fish? So I want to tell you why I care about fish. First of all, they're the most diverse group of vertebrates on the planet. There's more species of fish in the planet than there are amphibians, reptiles, mammals, and birds all combined. They're also dominant consumers in marine ecosystems. So if you go into the ocean, chances are if you're going to find a predator, it's going to be a big fish. Fish are also a really important protein source for much of the world. They provide livelihood for tens of millions of people and support a multi-hundred-billion-dollar industry, and provide protein for almost half of the world's population as the primary protein source. And so understanding how fish diversity came to be and understanding how fish interact with their ecosystem is absolutely essential for understanding and mitigating humanity's interactions with our oceans and with our resources in the ocean. And of course, the other reason that we should care about fish is that fish are cool. And I don't think that should be overlooked. I mean, come on, they are absolutely bananas in their morphologies, in their behavior, and their ability to live in extreme environments. And so I'm really excited about understanding fish from all of these different perspectives. But the other thing for me is that my background is in biological oceanography. And one of the things that I love about biological oceanography is that from a first order, we can take physical oceanographic conditions, things like temperature, salinity, pH, nutrients, ocean circulation, and broadly predict not only the primary productivity, the amount of primary productivity on the planet in different places, but also the amount of primary productivity and the amount of nutrient input and the temperature and circulation, all these other feature or abiotic factors of the ocean can contribute to our understanding and to the biodiversity and abundance of different kinds of plankton. So in regions where you have relatively low productivity, you might have a cyanobacteria-dominated system, whereas in regions with higher productivity, you might have a system dominated by diatoms or other larger primary producers. Of course, the diversity and abundance of plankton at the base of the food web will drive the abundance and distribution and diversity of the larger consumers in the ocean. So fish, sharks, whales, and things like that. And so from a first order, the things that are going on at the base of the food web or in the climate system can be driving changes in these predators, but also changes in these predators can potentially be indicative of changes in the climate system. And so to me, what's really interesting about the science that I get to do is I get to explore these relationships. Can fish tell us about climate, and can climate tell us about fish? So we're almost there. I think about fish as an ecosystem proxy. They can tell us about ecosystems. And for what we're about to spend the next few minutes exploring, I would like to talk about 3 different broad scale parameters that we're going to think about, or concepts. The first concept is production. So how many fish are there? Or more relevantly, how much energy is available at the base of the food web and then transferred up to the consumers? So when I say the word production, we're asking not necessarily how many fish individually are there at one moment in time, but we're asking how many fish or how much fish biomass is the ocean producing during an interval of time. In addition to the how many fish question, we can ask questions about the structure of the fish community. So those questions can be things like, who are the major players? What are their relative abundances? Do we have more big fish or more small fish? Do we have a higher abundance of sharks or do we have a lower abundance of sharks? How does that compare to other groups of fish? And so when I talk about community structure, we're looking at sort of this overall metric of what is the community looking like. In a lot of senses, this can be a pie chart or a ratio, or maybe a histogram of some kind of variable like size. And then finally, we can get to this set of questions about evolution. So that's questions about diversity. How many morphologies of fish are there? How much biodiversity are there? And is there? And how can we see when evolutionary events happen in relation to things like changes in the structure or the production? And so you can sort of see how each of these stories might give us different answers because we're asking different questions. There's how many total, there's how are they distributed, and then there's how many individual. And what those things look like can be potentially really, they can be very similar, they can follow the same trends, or they might be decoupled. And that's something that I'm really interested in exploring. So I don't have to tell you all that deep time becomes this really exceptional window into environmental and ecological change. I think about the planet and how many different transitions and changes it's undergone over its 4.5 billion year history. And we can actually use all of those changes to get a sense of what kinds of repeatable shifts can we see in response to environmental changes. So for the purposes of this talk, we're mostly going to be in the latest Cretaceous and in the Cenozoic. And so I just want to orient us all to this absolutely delightful figure produced by Thomas Westerhold and team that's looking at Cenozoic climate. And I just want to point out a couple of things. First of all, the early Eocene is very, very red. This is a bottom water temperature. This is a very warm climate in the earliest Eocene. We transition from a warm climate where we don't really have ice on Antarctica in the Paleocene and Eocene to a cool house climate in the Oligocene and Miocene that becomes the ice house that we see today. And the point that I have with this is that the climate has been variable, and we can use different changes in climate to look at changes in ecosystems and see how those changes either co-occur or don't, and whether there's similar direction and change across similar events. And so that brings me to this question of how has life responded to these changing climate and environmental conditions? And so that's what we're going to do. That's what we're going to look at. And the tool that I use most commonly is scientific ocean drilling sediment cores. And so this is just a little schematic that I made ages ago, where the drill ship, the JOIDES Resolution most recently, but there have been others as well, will basically collect a tube of sediment going down sometimes hundreds of meters into the seafloor, where the older you go, or the deeper you go, the older you get. And we have all sorts of records of basically anything with hard parts that gets preserved in these sediments. And for me, one of the most powerful things about scientific ocean drilling is that the program ran for over 55 years. And during those 55 years, they collected mud from all over the world. And although not all mud is created equal, all of these sediment cores are available and can be used for scientific research. And that's allowed me, as a relatively early career researcher, to be able to look at samples from all around the world and from all sorts of different time periods. And together, these sediment cores can give us not only a diverse reconstruction, global reconstruction of environmental conditions, generally using geochemical signals preserved in these sediments, but the microfossils that are preserved in these sediments can give us a record of life. And so between the geochemistry, environmental paleoproxy records and the microfossils, we have these absolutely gorgeous records that are paired records of life and climatic interactions. And what I'm really interested in is understanding those life reactions. And just for those of you who haven't spent your lives staring at ocean sediment cores, I throw this picture in here. This is my PhD advisor, Dick Norris, who is an absolutely fantastic PhD advisor, and I highly recommend getting to know him at some point. He is currently in this picture, he's on the JOIDES resolution in the North Atlantic Ocean, and you can see this little green wedge of sediment. Dick is very excited about this little green wedge of sediment because that is actually impact ejecta from the asteroid that slammed into the Yucatan Peninsula about 66 million years ago and bumped off the dinosaurs. And so I like this core a lot because it gives us a really visual reminder that we can actually, in these kind of sediment cores, look at before and after very specific moments in Earth's history. And that's a really powerful tool. So, all right, we've talked about time, we've talked about deep sea sediments. Now let's talk about my favorite fossils. So the fossil group that I have spent most of my career working on are ichthyoliths. These are microfossil fish bits. Ichthyolith literally means fish stone. And they are effectively a microfossil proxy for fish and for sharks. Ichthyoliths come, broadly speaking, in two flavors. There's the denticles, which literally means little tooth. Denticles are scales primarily found on sharks, but also found on skates and rays at much lower abundances. And they tend to cover the body of these elasmobranch organisms. Denticles are about 100 to 500 microns in diameter. And sharks can have hundreds of thousands of those denticles in just a small couple centimeter square patch of skin. And so, although sharks constantly are shedding their teeth, and those make an excellent fossil record, a shark might shed 10,000 teeth in its lifetime, maybe more, maybe less, depending on the species. It's going to have orders of magnitude more denticles from its body enter the fossil record than it ever made of teeth, just because of their size. In addition to the denticles, we also have teeth. Teeth can come from sharks and they can come from fish. Primarily what we see in the deep sea record are teeth from ray-finned fishes. And these teeth are triangular, and the vast majority of them are extremely, extremely small. My lab works with samples greater than 38 microns because I'm going to be honest here, that was the smallest sieve size that we had access to when we were doing this work. I'm quite sure that we're losing at least some teeth, but not a ton of them. But I will say that about 80% of teeth in the deep sea samples that I look at are less than 63 microns. So for those of you who work on forams and wash at 38 or at 63 microns, you're washing about 80% of my data down the drain. And so I encourage you to think about finding smaller sieve sizes or saving your fine fractions. But these fossils, they're pretty small, they're hard to get at, but if you have enough patience and enough effort, they are actually the most abundant vertebrate fossils on the planet and have the potential to tell us a ton about sharks and fish through time. So how do we get at them? I've spent a lot of my career trying to figure out how to get at these teeny tiny fossils. In a chunk of deep sea sediment, about 10 grams, we might have 50 fossils, we might have 500 fossils, but it's nothing like the tens of thousands of forams that you might get in a similarly sized sample. So I've developed a bunch of techniques for getting at these. One of my favorites is that we can dye the teeth pink, and that makes them easier to see and compare to silica grains, which don't pick up the dye. We've also been doing a lot of heavy liquid separation recently, which has been a really powerful tool. But ultimately what we do is we do our best to isolate the fossils physically and chemically, and then we finish the job off with a little teeny tiny paintbrush under a high-powered microscope. And then we take some pictures, and that's what we do. And then we can start asking these questions. Okay, we've got the fossils. What can they tell us? And now's the fun part. So I'm going to take us back to a very rough, should have been a Tuesday afternoon, very rough Tuesday afternoon, about 66 million years ago when the asteroid slammed into the Yucatan Peninsula and bumped off the dinosaurs. This is the Cretaceous Paleogene mass extinction. We talked about it just a minute ago. And this was a really bad day for a lot of organisms in the ocean. It's hypothesized that there was a collapse in marine primary productivity. We see a dramatic extinction in calcareous plankton. So there's a greater than 90% extinction in planktonic forams and in calcareous nanofossils. There's also a major extinction of a lot of large marine predators. So ammonites and marine reptiles were all victims of the extinction. And so what I wanted to do was understand, okay, well, the base of the food chain kind of went and the top of the food chain kind of went, but what about the things in the middle? What happened to them? How did they respond? And so what I'm going to do now is wake you all up. See if you can find that raise hand button in Zoom. You might have to find reactions or something like that because we're going to take a little vote. Uh-oh. I think maybe I just did a silly thing. Can you guys still see my screen or is it gone?
No, we can still see your screen.
Okay, beautiful. Sorry, Zoom did a weird thing to me, but I think we're back. Apologies. So I'd like you all to vote whether you think A is before the mass extinction or B. So give it a second and then we'll let you vote. Let's see. There we go. Zoom is better. All right. How many of you think that A is from before the mass extinction? One. I got 1, 2. Amazing. All right. Put your hands down. How many of you think B is from before the mass extinction? Excellent. Got a few more of you. Congratulations. You haven't fallen asleep. I really appreciate that you were paying attention. All right. Good job, bees. Thank you for that. It turns out that the A's are actually correct. A is before the extinction and B is after. And that's pretty counterintuitive. And actually, when I first started seeing this pattern, I thought that I had flipped my core upside down. I thought that I must have done something completely wrong. But it turns out that no, this is just a pattern that we see basically everywhere. And I promise you, I did make absolutely sure that I hadn't messed up my math and I hadn't flipped the cores upside down. And so there's a bunch of different things that we can observe in here. For one thing, the assemblage B has a lot more diversity in tooth form. There are also a lot more bigger teeth compared to assemblage A. But the thing that I actually noticed first is that compared to looking at A versus B, is that A had about the same number of denticles as it had teeth, whereas in B, we have a lot more teeth than we have denticles. And these are just some of the ways that we can try and quantify this, and so I'm going to show you some plots. So the first plot I want to look at is this one. This is just looking at now the ratio of teeth to denticles in one of these sites. And you can see that before the extinction in blue, it's relatively low. It's a little bit less than 1 to 1. But after the extinction in green, we see a big jump in the ratio of teeth to denticles. So there's about twice as many teeth as there are denticles. Now, I'm not saying that there's twice as many fish as there are sharks, because a lot more goes into figuring out the relative abundances of these things than just how many fossils they left. But certainly, after the extinction, it looks like fish are leaving relatively more fossils than sharks. We can also look at the size of these teeth, and here I've plotted in red the maximum size of the teeth in each of these assemblages. And although it's a little hard to see, on average, the Cretaceous maximum tooth length was about 1 millimeter. But once we hit the Paleocene, it's about 3 millimeters, with some exceptionally very large teeth that also show up. And this pattern that I observe in one site was actually true not just in the one site that we looked at, but all around the world. And so that was something really, really important and started to make me think, okay, what's going on here? And is this a mass extinction phenomenon or what? And so to dive a little bit deeper into this, I'm going to take you on a little vacation to the South Pacific Ocean to DSDP site 596. It's one of my favorite sites. It's a red clay site, so it's actually a little bit weird of a site. It doesn't have all of the carbonate samples that people are very familiar with in paleoceanography, but it has a fairly interesting set of data that helps us understand how old it is. It's got an inverse cobalt accumulation age model, and I'm happy to talk about that in Q&A if you want. But we also have a very well-developed iridium anomaly, which means that we can point at the moment the KPG extinction happened because we have this iridium layer that is where the fossil or where the asteroid hit. And so now what I'm going to show you is some production. So this figure is showing you things about community structure. So that's how many fish versus sharks. This figure here is showing us something different. It's showing us production, how many fish fossils are there and how many shark fossils are there. And the metric of production that we're using here is ichthyolith accumulation rate. And so that is how many ichthyoliths, or teeth and scales, are falling to the seafloor in a specific unit of time and in a specific area. And what this does is it allows us to normalize for changes in sedimentation rate. So what we're looking at is changes in production of the fossils rather than changes in overall preservation. And here we have, in black is total ichthyolith abundance, blue is fish, and red is sharks. And we're running from about 75 million years ago to about 40 million years ago. And if you squint, if you look between 75 and 66, you'll see that the red sharks are just above the blue fish, but at the extinction horizon, which is this dotted line, we see those switch. So we see a lot more blue compared to red, and that's that community structure change. But if I were to ask anybody here to tell me where a big change happened in this fish community, I would not pick the KPG extinction. I would actually look around 50 million years old to, as it turns out, the early Eocene climate optimum, which you all have maybe remembered I highlighted a little bit ago as the warmest time period in Earth's recent history. And so it turns out that at least in this location, we see this huge increase in fish abundance during this elevated interval of high temperature. And if we plot fish abundance compared to temperature, they correlate extremely strongly. And I've done some really interesting work with a colleague to try and figure out what might be driving this strong correlation between fish abundance and temperature. And we found that there's some really interesting things potentially driving this, potentially an increase in primary productivity during this time period, but also it may be that the ecosystems just became more efficient. And it turns out that we see this pattern of higher fish abundance with higher temperature, not just in the open ocean, but we also see it in the Southern Ocean as well. And so this leads us to this question. Okay, we've got a lot of fossils. We've got a lot of things going on in the early Eocene. How can we then take these production community structure things and turn them into some kind of question about species and species and evolution? So I'm going to talk to you a little bit. I thought I made a new figure for us. I'm sorry. I'm going to talk to you a little bit about determining species. How do we know what evolution is going on in the fish? And I get a lot of teeny tiny triangles, and I have a colleague who teases me that all I do is look at little ice cream cones all the time. But the thing is that if you stare at little ice cream cones long enough, you start noticing all of these potential differences. And so what I've been doing for the past many years now is developing a way of quantifying tooth morphology that is repeatable and that allows us to capture morphological variability of these deep sea samples without necessarily knowing what they are taxonomically, because it turns out that the deep sea record doesn't have a lot of really great on land body fossils that we can be like, oh, this tooth looks just like one that I have a solid taxonomic identity for it. We just have these isolated triangles. And so what we then do is we can take these morphological variations and create morphotypes. Now, a fish might have one morphotype of tooth in its jaws, or it might have 4 or 5. We haven't really found anything with more than that in it. But the number of morphotypes is roughly equivalent to the change in species abundance. And we've been doing this both for teeth And we've also been doing it quite a bit for denticles. And actually, the denticle paper is formally in press, so we're really excited about that. The other thing we've developed is a bunch of user-friendly guides. So if you too are really excited by tiny triangles, we've got an R package that allows you to do all of the analyses that we're doing, or that I'm about to show you in this data set. And it's a cool tool. We're trying to make it better. That's what we're up to. So happy to talk about that as well. Well, what we can then do is do things like look at morphotypes through time. And so this is kind of a complex figure. What it's showing you is a data set with 136 morphotypes, where each dot is representing a moment in time when that morphotype is existing. We can, for example, look at the origination rate of morphotypes in the Cretaceous compared to in the Paleocene. It turns out that there's more morphotypes showing up in the Paleocene than in the Eocene. Even though we have an order of magnitude more fish in the Eocene, there's not that many new morphotypes. That's pretty cool, and that means we can quantify that. I've been doing quantification of this in a bunch of different ways. I'm going to show you one of them today. This is capture mark recapture, which is a technique that allows you to look at both the presence and the absence of these fossils. And so here we're just looking at the abundance of fish at this interval. The red here is the estimated extinction. The blue is the estimated origination. And we can actually put error bars on this and see that there are big pulses of origination right after the KPG, but before we have high productivity. So you'll remember that I mentioned that there's sometimes decoupling between abundance and diversity. And this is a really good example of that, where the diversity peak came first and then the abundance peak came second. We're still trying to figure out why that is the case, but it's a really neat observation nonetheless. We can also look at their morphologies in a little bit more depth. And so one way to do that is to use all of those morphological characters that I mentioned, and we can put them into a ordination and figure out which teeth are more similar and more different. And so I'm going to show you a bunch of plots that look like this, where we've got dots, and the clusters of dots mean teeth are more similar. The dots that are farther apart means they're more different. This is an example of Cretaceous assemblages compared to earliest Paleocene and then later Paleocene. And then we have the Eocene. And this is a beautiful animation, but it's not super informative, except to say that most of the teeth fall within the central range. But if we look at more specifically times of origination and extinction, we can see that during different periods of this post-Cretaceous Paleogene boundary recovery interval, fish are existing in different parts of this ordination space, in different parts of this morphospace. And so during the Paleocene, when we see a lot of novelty, we're seeing expansion of that morphospace. But during the Eocene, when we see a lot of abundance, the occupied morphospace is relatively stable. And so this leads me to this conclusion that the Cretaceous-Paleogene boundary was actually a great thing if you happened to be a fish. And that's pretty cool. And that led me to this sort of one of the many lines of research that I've been up to recently. And I want to spend my last like 5 or so minutes talking with you about that. And that's this question of, okay, so we've got a lot of teeny tiny triangles. We can do some cool things with them. We can observe different patterns and processes. But that leads me to this question: who were these things? What made these fossils, and what did they do? And the short answer is it's really complicated. And I'm just pulling this little figure up for the purposes of today. Some of you may be familiar with the Elderfield proxy calibration, proxy optimism confidence curve. When I started this project and when I started working on fish teeth, I was like, they can tell us everything we ever wanted to know about fish and sharks. And we call that the optimism phase. That's this green dot. As we continued working on it, we were like, oh no, we can't actually learn anything from this. It's way too complicated. And that was the pessimism phase. But what's happening now is that as we're working through this pessimism phase of, well, it's just way too complicated of a system, there's too much convergence in the system, all teeth look the same, it doesn't matter what animal they came from, all scales look the same. We're starting to address the pessimism phase and get a lot more data and a lot more morphological data, and we're reaching what I what we think of as the realism phase, which is that this proxy can't tell us everything. It isn't telling us nothing, but it can tell us some things. And what I want to do today is share one of the things that I think is telling us that is pretty robust and pretty exciting. So how do we do this? Well, first of all, we don't have a lot of Cretaceous and Paleogene sharks and fish fossils that are from the deep sea, from the samples that I'm looking at, that can tell us, oh, this tooth definitely came from this species. So what we've been doing is taking a modern calibration approach, looking at all of the modern sharks species that we can get our hands on. We're looking at shark scales across the body to get an idea of denticle diversity. So this is just an example of two different sharks with two different kinds of denticles. And you can see some of them have very similar denticles across their body. Others have more diversity. We've been collecting samples from, again, all across the shark tree of life to look at denticle diversity, and then trying to use that to compare to what our fossils look like. We've been doing the same thing with modern fish. And there's about 33,000 species of these, so we're working our way through it. We started with taxonomic breadth, trying to get some of every order, and now we're into the sum of every family. But every now and again, we find something that's really cool, and we can take a taxonomic deep dive to really better understand some feature or some group of fish. And so the last little bit that I want to share with you is a taxonomic deep dive into a group of deep sea fish called the stomatiiforms or stomiiforms, depending on who you happen to be talking to at what moment of fish taxonomy. And that is this little group down here. And this project that I'm going to share with you is inspired by a really great conversation I had with Carly Cohen, who is at, actually, she's at University of Washington now. I need to update that. She just moved. She's a postdoc there. And she's perhaps the only other person in the world who cares quite as much about tiny fish teeth as I do. She's a modern fish tooth morphologist. And she and I were chatting about different tooth morphologies, and I mentioned these spiral things, and she was like, oh, I think I know, maybe we've got some spiral things too. And she showed me a picture, and I was like, that's not the spiral I was talking about, but I've got those too. And it turns out that there's this fish in the ocean. You've probably never heard of them, but they are the coolest little critters you've never heard of. They are a group of fish. Their genus name is Cyclothone. They're about 5-10 centimeters in length, and they are the most numerically abundant fish genus in the ocean, which also makes them perhaps the most numerically abundant vertebrate genus on the planet. They live primarily in the deep sea, but they do vertically migrate, and they have these absolutely gorgeous, highly distinctive spiraling teeth. And they're quite tiny. They're about 50 microns in diameter, which means these are some of those teeth that euphorium folks will wash down the drain if you're washing with a 38 or with a 63 micron sieve. And it turns out that the oldest body fossils known of Cyclothone are from the Middle Miocene, about 12 million years ago. And so I had these fossils and I was like, are they actually Cyclothone or not? And so what we did was we decided, okay, well, they might be Cyclothone. Certainly we have a couple of pictures of different Cyclothone, and they all seem to have these spiral teeth, but we didn't have any data from other groups in the related parts of the tree. We did go through the entire catalog that I had of, it was about 300 fish species across all of the orders at the time. And Cyclothone was the only one who had teeth that looked like this. But that wasn't good enough for us. So what we did was we recruited Corinne Tenenbaum, who is an undergrad at Yale. And we had Corinne image every single specimen of stomatiiform fish in the Yale fish collections. And that was a lot of fish. She did a fantastic job. And what we found when we were looking at this is that in the stomatiiforms, a lot of different teeth had textures compared to most of the rest of the fish teeth in our collection that were very smooth. And so we started to try and define these textures, and we defined them. We've got a number of different surface textures that we could see. And then we decided that we were going to plot those on a phylogeny. And this is what you're looking at. This is a phylogeny of stomatiiforms downloaded from the Fish Tree of Life. So I believe that's the Roboski tree from 2018. And we've just plotted on here an ancestral state reconstruction of different surface textures. And we found a bunch of really cool things. One of them is that Cyclothone have spiral teeth, and they are the only ones that have that spiral texture. Super cool. It also seems like there are some other really distinctive surface textures that are taxonomically constrained within the context of this group. And we also note that the vast majority of fish teeth are smooth. And so it may be that surface texture is a stomatiform specific feature, or it may be that there's some others out there somewhere that we haven't yet sampled. But for the time being, we think that we're pretty confident, at least, that these cyclothony teeth or the cyclothony spiral teeth could be indicative of what's going on, of a real taxonomic identification. And so that leads me to the fossils that started this all. So A and B here are two different fossil teeth that we think have the same kind of spiral striation and little curved features that we see in the modern cyclothony. There's two species of cyclothony here in C and D. These are modern teeth. You can see they've got the same curve, about the same size. They're all about 50 microns in diameter, and they even have that cute little cap tip. And I told you at the beginning of this whole thing that cyclothony's oldest body fossil was about 12 million years old. But it turns out that our oldest fossil so far from the deep sea is actually about 56 million years old, which suggests that this clade really has had a presence in the open ocean for perhaps 40 or 50 million years longer than we thought it did. And that's really cool. And that's just one of these like proxy optimism things that I'd like to leave you with. So I promise I'm wrapping up. I just wanted to say thank you again for having me. Thank you for continuing this really cool PaleoParks legacy. I hope I've convinced those of you here still here that ichthyolites are pretty awesome and that they can provide all sorts of insights into productivity, community structure, and diversification of fish and marine ecosystems. I hope that you come away with this thinking that maybe the Cretaceous Paleogene extinction wasn't so bad if you were a fish. And the things that I'm working on now are trying to be able to do this widespread taxonomic and potentially ecological reconstruction to be able to put more biological information onto all these little teeny tiny triangles and little tiny beautiful intricate sampling or little intricate fossils of denticles. So with that, I would like to say thank you to everyone who's been involved in this set of projects over the many years. As a new faculty member, it's always a privilege to be able to continue this work and expand it and share it with other folks. And so my lab group, we are the Paleo Fishes Lab. It's actually a real acronym. And so we're really excited to be able to do this work, and hopefully we'll be able to continue for years to come. And so with that, I will take any questions you've got left over.
Thank you for that great talk, Elizabeth. I really enjoyed it. And also, as part of the current PaleoParks committee, thank you for helping setting it up and laying the groundwork for what has been a very successful seminar series, I think. So yeah, with that, let's go to the questions. Reminder for everyone in the audience, if you have any questions, you can send them to the questions at PaleoParks host. We already have a few, so I will both post them in the chat and read them out loud. The first one is from Kartik Sendura, and he asks, how does the accumulation, i.e. the ichthyolites per million years per area, accommodate for variable current activity? And are ichthyolites from a particular depth more prone to preservation?
Both of these are really great questions. So I guess the variable current activity, you're thinking about like ocean currents moving around. And then I guess oceans from a particular depth more prone to preservation. So two things. One, if there are currents in the ocean that are moving things around, there's actually been some really elegant modeling looking at how sinking of particles, mostly forams, but also marine snow and other things like that, how sinking particles interact with ocean currents and how that moves them around. And in general, for open ocean settings, what's happening in the surface is basically making it down to the seafloor, within a relatively reasonable radius to think that what's going on in the water column is being preserved. In terms of ichthyolites from particular depths being more prone to preservation, we're thinking like fish living at the surface versus fish living in the mesopelagic versus fish living at depth. We aren't 100% sure how ichthyolites get to the seafloor. But we can make some guesses about this, and I promise we'll get to the question. If you're like in a lake and you're a fish and you die and you sink to the bottom, usually there's a chance that you get covered as a whole body. In the open ocean, if you're a fish, the likelihood of you dying by like a flood or a landslide or something is very, very low. The likelihood of you dying by being eaten by something, however, is incredibly high. And so most of the death that happens in fish in the oceans is because they got eaten by something. And it turns out that fish teeth are pretty resilient and they tend not to dissolve even in simulations of fish guts. So one of the projects that I've done is simulating different time transits in simulated fish guts to see if we can dissolve teeth and what preservation that looks like. So we think probably how most of these things are getting to the seafloor is that a fish gets eaten and then it gets pooped. And that fecal pellet then sinks it down to the seafloor relatively quickly. But a lot of things in the deeper ocean are also eating marine snow as it comes down. So it's kind of getting cycled through multiple fish guts on the way down. In terms of preservation, if you're up in the epipelagic in the surface and you get eaten and pooped, you might go through several cycles. Whereas if you're in the mesopelagic or the deep, the bathypelagic or even deeper, you're probably going to go through fewer cycles of that. But I don't know that we have any real data about preservation bias based on sample or based on water column depth. Like I don't think anyone's done that kind of experiment. So that's my guesses.
Thank you for that very comprehensive answer. There is one question in the chat by Brian, who he asks, how do you normalize for varying sediment accumulation rates and sample size at different locations for estimating ichthyolite diversity and abundance through time? Sorry, I didn't read that completely correctly, but I think you can figure it out.
Yeah, that's a great question, Brian. So when we're doing the ichthyolite diversity through time estimates with capture recapture, we can actually include things like covariates in those analyses. And so we can actually, one of the things that I look at in there, or one of the covariates I have is basically sample abundance, like how many fossils are in this sample. And that allows us to get an estimate of origination and extinction that is accounting for, oh, well, if we've got a really small sample, then it's probable that our detection probability is lower in this sample, and that can then be backed out with a maximum likelihood framework to figure out what, if a detection probability is lower, what is the overall extinction event or extinction or origination estimate. And so those are covariates that can go into the analysis and they end up as part of the error bars as well. In terms of estimating abundance through time, sediment accumulation rates, we normalize ichthyolith. So what we can collect is the number of ichthyoliths per gram sediment. But if you have higher sedimentation rate, you're going to have fewer ichthyoliths per gram sediment for the same number of teeth. And so we normalize to sedimentation rates. So it's just ichthyoliths per gram times sediment density is grams per centimeter cubed times sedimentation rate is centimeters per 1,000 years. And that gives us an ichthyolith accumulation rate. And that ichthyolith accumulation rate is, it's taking into account the changes in sedimentation. Both sediment type and sedimentation rate.
Thank you. We have another question from Gwen. She asks, can you talk about the applicability of your image processing techniques to other kinds of microfossils?
Yeah, that's a great question. So right now, the image tools that I'm using, primarily I've been using Automorph, which is a software developed by Allison Sheng and Chelly Hull. And it's a great piece of software that allows you to find individual objects, usually light objects on a dark field. It was primarily developed for forams. I've adapted it for fish bits. We have to change some parameters because my fossils are a little bit shinier and also a little bit more see-through than forams are, which means the image parameters are quite a challenge sometimes. But we can definitely look at, or we can definitely use that tool. one of the things that I ended up struggling with, especially early on in this work, is that a lot of the image tools then take the next step of finding an outline of an object and using that outline as the primary morphological data. And I learned really early on that that was not going to be an acceptable or appropriate way to analyze the vast majority of my fossils. Because these things are so tiny, especially the teeth, getting a tooth perfectly oriented flat on a slide to be able to have a repeatable outline was nearly impossible. And so a tooth that was perfectly flat and a tooth that was the identical morphology, but just slightly tipped, was giving us entirely different outlines. And that's how we ended up with this set of discrete characters that we work with, because the fossils were just so small that we couldn't do a good job of that, of it just using traditional outlines. Now, I'm actually in the process of working up a really cool machine learning technique that's using basically a bunch of machine learning algorithms in parallel to get hybrid morphological codes. And hopefully in the next couple of months, I'll have something really cool to show with that. And that would actually then become very applicable to other folks who are trying to do morphological based analyses where you have many bins that you want to fall into, but maybe not enough training data to train on thousands of bins. And so that's something that, again, we're working on, and we've got some cool things in the pipeline, but that's potentially quite applicable to other groups.
Thanks a lot. We have another question from Carl in the chat. He says, great and accessible talk. And he asks, are you aware of any general correlations between ichthyolite and planktonic foraminifera, abundance or diversity?
Also great question. So today I presented some KPG work, specifically because although it's a little bit older, I can talk about all of the different metrics from one location. That makes it a nice story to wrap up together. It seems like ichthyolite accumulation at the KPG tends to recover before foram accumulation. But then if we look at the timing of ichthyolite diversification after the KPG, that seems to line up with specific diversification events, first in calcareous nanos and then second in forams. And so sometimes it seems like they're decoupled, but sometimes it does seem like we've got some events that are sort of right next to each other. The other point that I could make briefly is that I didn't talk at all about the early Miocene work that I've been doing, trying to understand what happened to marine ecosystems during that interval. And we find a major extinction in sharks across like a weird interval in the early Miocene. There's also a huge change in planktonic foraminifera communities across that interval. It's not necessarily planktonic foram diversity, though there is a shift in diversity, but there's a huge change in the co-occurrences. So like the who is living with who in planktonic forams has a huge shakeup during that time interval. The only other shakeups like that are at the EO boundary and at the KPG for the forams. So it's a really big deal in the forams community. And so there's some interesting things going on there as well. And again, it's one of these questions of what metric are you thinking about? Is it how many species? Is it what's the community structure? Is it how much abundance is there? Is it yes to all of these? And I think that's a point that I really learned as I've been doing this, is like, it matters not just what question you're asking, but how you're going about answering it.
Thank you again, Elizabeth. We have two more questions, I think, and then we're close to the top of the hour. So the first one is from Kartik Sandra, and he asks, are some fish tooth textures more prone to abrasion than certain other textures? And what does it tell us about the niche occupied by the fish?
Great question. We think that most of these surface textures are actually really, really tiny. So we're talking order of like 5 to 20 microns in size. And so that's actually not particularly prone to abrasion just because of how tiny they are. We're still trying to figure out what they mean. We've got some experiments that we're starting to do, trying to do some functional morphology to see if surface texture has something going on there, but I don't have a good answer for that right now, unfortunately.
Thanks a lot. And then I think we have the final question by Moritz. First, he says, thanks, Elizabeth, fantastic work. I wondered, have you tried other ways to characterize and differentiate ichthyoliths? For example, different stains, micro-XRF scanning, or paleoproteomics? If so, what did you find?
Excellent question. So we haven't done a lot of, I personally haven't done any paleoproteomics because the samples that I'm working on are so old and also just so tiny that there's not enough material to do that. Maybe sometime in the next 10 to 20 years, we're going to get there. But at this point, we're talking about things that are like, 100 microns in size and just don't have any genomic material. In terms of micro XRF scanning, I haven't personally done any elemental mapping. I don't have access to that kind of instrument. And in different stains, we do use alizarin red S sometimes to stain the teeth. But these things have been dead for so long that other kind of biological stains aren't giving us real information. So right now we're just using reflected light microscopy and it's doing the trick most of the time. But I think there's a lot of potential. One of the things I'm really excited about is single tooth oxygen isotope measurements because different groups of fish live in different parts of the water column. And different parts of the water column have totally different temperatures. And so there's the possibility of reconstructing thermocline dynamics from deep sea fish with single tooth oxygen measurements if we can get good enough measurements out of them for that. So that's something I haven't done a lot of because we haven't been able to get the standards good enough to do it. We keep trying, maybe we're getting closer. But I think that's another example of ways to characterize and differentiate that we're excited about.
Thanks a lot. With that, I'm going to close the Q&A because we're at the top of the hour. So thanks again, Elizabeth, for the great presentation and for answering all the questions. If you have any more questions, again, please stay for the tea time. I am briefly just going to share some logistics and info for the next talk. Then we take a quick break, and then we'll have tea time. So yeah, again, thanks everyone for joining today's seminar. Again, please feel a reminder to fill out the weekly feedback form, which will again be linked in the chat. We really would appreciate that. We don't know if we're going to have a seminar next week, but we definitely hope that you will join us on May 27th when we have a talk by Pam van Voort, who is going to talk about paleoclimate modeling. So for those that don't want to stay for tea time or are not able to have a great rest of your Tuesday. And for those that will stay, we have tea time next. So tea time is just an informal chat where you'll be able to unmute yourself and turn on your camera and just have a chat with today's speaker. And we always have some kind of icebreaker question to do that. And the question of the week is, what is your favorite fish, paleo or modern? So you can think about that in the 2 minutes that we are going to have as a break now. So you can have a quick drink of water and come back in 2 minutes. If you have any pets, we'd always love to see them. So bring them with you on camera. And with that, I hopefully see some of you in 2 minutes.